We're going to start by taking a closer look at the Neoproterozoic, which is the very last time in the Proterozoic. Basically, it ends right here at the end of the Ediacaran, which is where the Phanerozoic starts, around 542 million years ago. And it goes all the way back to 1,000 million years ago, which of course is the same as a billion years ago. And that's the Neoproterozoic. Now, we really only care about the last bit, the Ediacaran and up. The reason for that is that, well, it's the most recent, of course, but it's also the time after which we have a substantial thawing. This period just before the Ediacaran, the Cryogenian, is called the Cryogenian because it was really cold. We had a couple of major glaciations, the Sturtian and the Marinoan. And yeah, we have fossils from before that. The latest part of the Tonian is when we get things like Otavia, the earliest thing that might be a sponge from fossils in Africa. But you have to remember that the Proterozoic goes all the way back to 2.5 billion years ago, right after the Great Oxygenation Event. And so when we talk about these periods of glaciation in the Neoproterozoic, we're really not talking about the same thing that we discussed earlier. I don't know if you remember this, but we said that after the Great Oxygenation Event, there was a snowball Earth period from around 2.4 billion years to 2.1 billion years, which was the result of methane gas being replaced by carbon dioxide as a result of the introduction of oxygen. This is not the same phenomenon. There are probably other causes, probably related to the orientation of continents, that were responsible for these and other glaciations during the Neoproterozoic. So I'm going to redraw this timeline for us horizontally. Tonian, Cryogenian, and Ediacaran being the last part of the Neoproterozoic. The only time you really need to know by name is the Ediacaran because it's a time in which we have a lot of interesting stuff going on. And the real reason I want to do this is to remind you that right between the Ediacaran and the Cambrian, which is the first time of the Phanerozoic, we have the beginning of the fossil record, the well-known fossil record. And in the first time of the Cambrian, we have already established the major phyla that we talked about in the previous video. Chordates are there. Echinoderms are there. On the protostomial side of things, we've got mollusks, annelids, and flatworms, which are the Lophotrochozoans. And then let me extend this a little bit further. And then we have the arthropods and the nematodes, which are the ecdysozoans in the protostomial lineage. Okay. By the time we get to the Cambrian, all of these animal phyla, all these lineages have pretty much already established themselves, which basically puts their origins and their diversification sometime probably before the Cambrian, probably sometime during the Ediacaran or before. And so the presumed diversifications took place. Basically, if we have the chordate lineage coming across the Ediacaran-Cambrian boundary already established as distinct from the echinoderms, that means that sometime before this, I'll draw that with a dotted line, there was the split, the cladogenic event. Who knows exactly how long before it was. We have an undisclosed amount of time from the time that we actually had the cladogenic event that split the lineages of the deuterostomes. Okay, so right here you would say this is going to be our deuterostome common ancestor, or deuterostome MRCA, which stands for most recent common ancestor, right there. Okay. Similarly, if you look at either one of the protostomial groups, we've got mollusks, presumably coming across the boundary as distinct from the annelids, which come distinct from the flatworms, also known as a phylum Platyhelminthes. We know that the flatworms are probably the most basal group, and the mollusks and annelids are more closely related to each other. So we know the topology is going to be something like this, where these guys are sharing a relatively recent common ancestor compared to the common ancestor shared between them and the flatworms. But we really don't know exactly how long in the past, into the Ediacaran or even before that, where we've got the ancestor to all the Lophotrochozoans. Lophotrochozoan merca. All right. And similarly, of course, we have the arthropods, the nematodes. They make it across the Ediacaran-Cambrian boundary, presumably already distinct from each other, and their common ancestry through an ecdysozoan merca is sometime in the past. And going even further into the past, we realize that the Lophotrochozoan and the Ecdysozoa are both protostomes. So we've got another unknown period of time that transpires between their common ancestry and the time that they split into their respective lineages. The Lophotrochozoan ancestor splits in the mollusks, annelids, and flatworms at a later point relative to where it split away from the Ecdysozoa. So the protostomial common ancestor would be here, and some point before that, you would say there had to be a common ancestor that connects all of the bilateria together. We can call this protostome, deuterostome common ancestor. Sometimes we refer to it as the Ur-bilaterian, using a term that comes from the German. Now, this period of the Neoproterozoic, extending backwards in time from the start of the Phanerozoic, is of interest to us because all of these events, all of this diversification had to have taken place prior to the start of the Cambrian, but we don't really know how far into the Proterozoic it extended. It could be extremely deep, it could be relatively shallow. We know that sponges are making their first appearance during the Tonian. We have our doubts about how much activity could be happening during the Cryogenian because it was just so cold. The Ediacaran seems like the best candidate period in which we could have had most of this diversification taking place. But the big question marks are these question marks back here. We don't know what these ancestors look like because there's no fossils. We don't know how long ago they existed because, again, there's no fossil evidence. And so this sets up the next part of our discussion, and I want to address two things specifically. The first one is, how long ago did these splits take place? How could we even begin to investigate that idea, given the fact that there's an absence of fossils? Because if there's a clear fossil protostome that's distinct from a deuterostome, we could say that the split between protostomes and deuterostomes right here would have occurred no later than however old that fossil turned out to be, right? We don't have any of those constraining fossils. And so the big question here is, well, what sort of information can we garner from modern organisms to allow us to estimate these divergence times, even though we don't have any fossil evidence? Okay. The second thing we're going to be talking about is, again, inferring from modern organisms. What did these animals look like? How could we use modern organisms to inform us about how these organisms might have existed, what they looked like, what they did? All right? So let's think about the first question. We know that during the Cambrian, we had the ancestral mollusk. Let's say the molluscan merca, for example, was there during the Cambrian. We don't have different kinds of mollusks. We know that from around 500 million years ago, we had a mollusk, a generic mollusk, and it split into two clear lineages so that by the time we got to the end of the Cambrian, we had one lineage where there was a cephalopod lineage as distinct from the bivalve lineage, which has subsequently had a long history that leads us ultimately to modern squids. And on the bivalve side, it leads us to a modern clam. Okay, now from our early, now from our earlier lab on phylogeny reconstruction, we know that it's relatively easy to go into GenBank and find comparable sequences between squids and clams, look at the nucleotide sequence for these two animals, and count up the number of differences there are. How many letters are there differences between the two sequences as opposed to the similarity? We could call the number of differences the genetic distance. So let's say we've got between modern squids and modern clams 30 differences. Now, without a whole lot of further thought, you might be able to also infer that if we were to look at other kinds of cephalopods, maybe an octopus, and other kinds of bivalves, maybe an oyster, if you were to compare an octopus with an oyster, they share the same most recent common ancestor that's shared by a squid and a clam. And so if the rate of molecular evolution is relatively consistent, then the number of differences we would find between an octopus and an oyster should be about the same as what we would find between a modern squid and a clam. So making a prediction about the number of differences between an octopus and an oyster, you would say that if molecular evolution occurs at a relatively consistent rate, you'd have about the same number of differences, about 30. And you'd be right. Turns out that molecular divergence rate for a particular gene stays relatively consistent within taxa. Now we can look at another lineage. We can look at another lineage like arthropods and say arthropods, similar to mollusks, have their most recent common ancestor documented for us by the fossil record at around the same time, during the Cambrian. And so from this most recent common ancestor, arthropods split into a couple lineages. One gives rise to like crustaceans, and the other lineage might give rise to insects. And you can follow these lineages out to modern times. Along the crustacean lineage, you could say, well, you would have a modern lobster as a representative crustacean, and you could have a fruit fly as a representative insect. And so we can make another prediction here. You can say that if molecular sequence divergence rates are consistent between taxa as well. In other words, if mollusks diverge at about the same rate as arthropods, then you'd also predict that comparing a lobster with a fruit fly, we'd have about 30 differences between those. And again, you'd be right. The data seems to support the contention that molecular rates of evolution are relatively consistent, at least within protostomes. It changes a little bit if we start looking at vertebrates. Vertebrates have different rates of molecular evolution compared to invertebrates. But as long as we're talking about invertebrates like mollusks or arthropods, it seems like there's some support for the idea that molecular evolution rates are pretty consistent. Now, if you were to look within insects, okay, insects, like for example, if you were to compare fruit fly with a cockroach, you would say that fruit flies and cockroaches diverged from an insect common ancestor more recently compared to the ancestor that either of these insects shares with Crustacea. So you would predict that there'd be fewer, far fewer than 30 differences between those two organisms. And again, you'd be right. And so there's a substantial amount of evidence from independent studies supporting the idea that molecular divergences can actually be used as a kind of chronometer or clock. A molecular clock is the term that was used by Moto Kimura, who discovered this. And I hope you can see where I'm going with this, because if 30 differences represents about the time between modern day and 500 million years ago during the Cambrian, then we would be able to use the number of differences between, for example, modern clams and modern lobsters. We should be able to compare sequences, a homologous sequence between a clam and a lobster, and get an estimate for the amount of time that's transpired since they shared a common ancestry way back here, right? Okay, so the time from here to the present, the time from right here to the present, should be proportional to the number of differences between the lineages that share the common ancestor at that point, in the same way that modern squids and clams share 30 differences corresponding to common ancestry during the Cambrian, right? Moreover, you could probably expect, you would predict that the number of differences between clams and fruit flies would be about the same. You would make that prediction, or lobsters and squids, or fruit flies and squids. You could say, well, any one of these four comparisons, all these four comparisons should be about the same in terms of the number of differences, and you'd be right. And the degree of distance beyond the 30 should give us some insight as to the amount of time that it's transpired from between modern times and this time way back here. I like to present this because it's a really nice example of how we've been able to make innovative use of molecular sequence information. We basically have this new information. How we're going to be able to use it, this seems like a good application. Now, I'm sure you're expecting me to tell you exactly how long ago this protostomial ancestor existed, but the truth of the matter is that we still don't have a good answer. It turns out that molecular analyses, for all their glories, I mean, they've proved to be incredibly dependable for some applications, like the reconstruction of phylogenies. This particular method of using molecular divergences to estimate the time of divergence is less than convincing. This methodology tends to give us estimates that are too deep in the past to be reasonable. For example, these estimations would give us a divergence time that goes way too far in the past to be reasonable. There's really no way that this event could have preceded the Tonian, and that's about where this estimation lies. And so the bottom line here is that I kind of built up a story kind of promising that there'd be some final resolution, where the reality is that this method, even though it seemed to be reasonable, turned out not to be particularly fruitful. Okay. Now the second question I wanted to ask, and spoiler alert, we're not going to come up to any distinct answer here either. Ask the question of how we could use modern organisms to recreate a picture of what this protostomial deuterostomial ancestor might have looked like. And our focus at this point is going to be on homologies, characteristics that are held in common by some highly derived protostomes. A highly derived protostome would be for us a fruit fly, as well as a highly derived deuterostome. And we've got a couple of modern deuterostomes that are kind of like favorite study organisms. One of them is the zebrafish, and another modern chordate that is used in a lot of laboratory studies, is a regular old house mouse, Mus musculus. So the idea here is that if we can find characteristics that are there in a fruit fly and are also there in a mouse, we could infer that that characteristic would also had to have been back here at the protostomial deuterostomial common ancestor, where both those lineages diverged from a single animal species. So let's make a list of characteristics that are shared between mice and fruit flies and zebrafish and fruit flies. Well, one is a master switch for eye development. Basically, this is a Hox gene. It's called iris in fruit flies. It's called PAX6 in mice. And in both of these species, if you don't have this gene, you basically develop without an eye. And not only that, it's the same gene. You could take the gene out of a mouse. You could use the mouse copy of the master switch for eye development called PAX6 and implant it in a fruit fly, and it can direct the development of a regular fruit fly eye in a fruit fly. And so there's been experiments to demonstrate that there's clearly homology between the fruit fly eye development gene and the mouse eye development gene. And so this would be a characteristic that's held in common by the two species. Another one of the genetic commonalities held both by, in this case, it would be fruit flies and zebrafish, is a master switch for the development of a heart. In fruit flies, this is called tin man because the baby maggots that were lacking this gene were developed normally except for the absence of a heart, and then they died. And so this tin man gene we learned was extremely important, was the master switch which triggered the development of a heart. And we found that this same gene was present in zebrafish and active during the period of heart development and in the same location where the heart develops in fishes. Again, really strong demonstration that it's the same gene that causes the development of a heart in fruit flies as well as in zebrafish. And there are a total of 6 of the Hox cluster genes out of the 8 that we find in fruit flies that are pretty much exactly the same as what we find in vertebrates, in mice, for example. HOX1 is the same as Proboscipedia. And so there's a total of 6 genes that are the same in fruit flies and in mice, which basically suggests that the ancestral protostome deuterostome ancestor, the Merca of the protostomes and deuterostomes, had all of these characteristics. It had to have had 6 Hox cluster genes. It had to have had the gene that we now identify both in fruit flies as well as in zebrafish as the gene that causes heart development, as well as the gene that we now recognize as the one responsible for the development of eyes both in fruit flies and in mice, right? Which leads us to a really interesting picture of this ancestor to all protostomes and deuterostomes. It must have been probably very worm-like in structure because that's essentially what a bilaterian looks like. But it presumably had eyes as well as maybe a heart was circulating the material inside. Okay? And not only that, apart from eyes and a heart, it presumably also had 6 Hox genes, which is kind of crazy. I mean, to say that you've got 6 Hox genes for a modern organism suggests that you've got a pretty high degree of body complexity. There are 6 different regions along your body axis that require distinct signals for appropriate development. And so this emerging picture of what the Ur-Bilaterian looked like is one of pretty high sophistication. And this totally does not match up with the established picture of what we thought this ancestor must have looked like, which is also based on extremely compelling grounds. Okay? For one, we would expect simple to be there at the origins, not complex. Right? Not only that, because this was happening fairly deeply within the Proterozoic at a time before there was oxygen and a time during which we have no fossils, it must have been extraordinarily small, probably less than a millimeter in length. If it were any bigger than that, then it would probably have left some fossil traces. And the third reason why the original bilaterian could not have been this complicated is that it really at this point hadn't even decided which end was its front and which end was its back, or for that matter, which side was up and which side was down. And here's where we get to one of the essential pieces in any lecture in a Bio 202-like class, and that's protostome-deuterostome differences. How are these two major lineages of bilaterians different from each other? Okay, and long answer is there's a long list. There are characteristics of cleavage. There are characteristics of how the mesoderm forms and how the coelomic cavity or the body cavity forms. I don't find it terribly productive to list those things. The one bit of information that's actually relevant to our current narrative is actually the one that gives us the two names of lineages, protostome versus deuterostome. And it kind of goes like this. Okay, now I'm going to draw out for you the basic structure of the gastrula. I showed you a picture of this before. Gastrula. And so you might remember that there's this general opening called the blastopore, and you've got the outer layer, and you've got an inside layer. The inside layer was the endoderm, and the outer layer was the ectoderm. And the big difference between the protostome and the deuterostome has everything to do with what happens to the blastopore. What is the fate of the blastopore? Okay, and so the way development proceeds after gastrulation is going to be different in protostomes and deuterostomes. Now in the protostome, the blastopore is going to become the mouth. Okay, so if I were to say this is a fruit fly, and this gastrula is eventually going to develop into a fruit fly. Let's say, well, here's the eyes of the fruit fly. Here are the wings of the fruit fly. And here's the abdomen of the fruit fly. The legs. Okay, and basically the second opening. Okay, there might be a second opening, might be the same opening, but basically I have a second opening on this side, which becomes the anus. But the blastopore itself becomes the mouth. Right? And this is completely different relative to what happens in the case of a deuterostome. In the case of a deuterostome, the blastopore is going to become the anus. Right? And so if this were, for example, a fish, okay, a fish would have a mouth on this side. Here's the eyes. Here's the gills. And maybe there's going to be a tail over here. You see the fish? And so in the case of a fish, the second opening, which is going to create a complete gastrointestinal tract, the second opening is going to become the mouth. That's where the term deuterostome comes from, second mouth. And so protostomes and deuterostomes are actually built on completely different body plans. Basically, what happens to the gastrula in its subsequent development is dramatically different. It's completely reversed in the case of protostomes and deuterostomes. I actually should have drawn one of these two things upside down, because it's also true that other aspects of body polarity, the dorsal and ventral structures, are entirely reversed in deuterostomes and protostomes. In protostomes, for example, the spinal column, the spinal cord, is along the ventral surface, and the digestive system is along the back. Whereas in the case of the deuterostomes, the central nervous system is usually along the back, whereas the digestive system is more ventrally located. Okay, so yeah, protostomes and deuterostomes are actually quite different, which suggests that the ancestor, this ancestor right here, the little worm that gave rise to the protostomes and deuterostomes, it can't have been terribly sophisticated because it really didn't even know which side was its mouth and which side was its anus, right? And so if you sort of visualize that little mouth, that little animal, little worm that had two openings, yeah, it had two openings. Basically, it had it had an archenteron, and one of these openings was the blastopore. And the second opening gave rise to a complete tract. We didn't know whether it was going to be moving this direction to become a protostome or it was going to be moving this direction to become a deuterostome. This had to have been a very simple little animal. The ancestor that we picture based on this embryological analysis, as well as the fossil evidence suggesting that this could not have been any bigger than 1 millimeter in length, is really at odds with the idea that we had this degree of sophistication and this degree of complexity. So yeah, there's no final resolution to the story, but there definitely is a lot of intrigue. And definitely there's going to be a lot more information coming your way as you continue with your biological studies. All right, so irrespective of whatever that was that guy looked like, he certainly did have a lot of offspring. He certainly did have a lot of success. If we put the Ur bilaterian way back here, and remember that's the protostomial deuterostomial common ancestor, you've got two major lineages, one being protostomial and the other being deuterostomial. And from a protostomial common ancestor, we have the splitting of two major lineages, one being the Lophotrochozoa and the other being the Ecdysozoa. And the Lophotrochozoa is a particularly rich superphylum. There are actually many phyla that are contained within this group, but we're going to be focusing on only three, one of which is going to be the Platyhelminthes, and the other two are going to be the phylum Annelida as well as the Mollusca. Now the Ecdysozoa is similarly a superphylum containing several phyla, but the two that are really important are going to be the arthropods and the nematodes. And I'll go ahead and preview the two phyla of the deuterostomes that we will be focusing on today, one of which is going to be the phylum Echinodermata and also the phylum Chordata. But we're going to be focusing on chordates in the next lecture, so I'm just going to briefly mention them here. Okay. We'll be detailing them later on. I'm going to be putting a few names on the board to kind of give us some anchor as to how to recognize or what kinds of organisms we're talking about within each of these phyla. And so this is going to be deliberately brief because, well, the way I see it, we could spend a lifetime talking about all these different kinds of invertebrates. At this point, I just want you to become functionally literate with regard to the identities and memberships of these different groups. Okay, so let's start with the phylum Platyhelminthes. There are several free-living flatworms, like the ones we are probably going to be seeing either in a marine collections or at the Birch Aquarium. We have parasitic forms of platyhelminths like tapeworms, as well as internal parasites like parasitic flatworms. I don't mean to imply that tapeworms are anything less than parasitic, but we've got a couple of substantially different forms of life, different body structures between tapeworms and the rest of the platyhelminths. Okay. Now the general theme for the platyhelminthes is that they've got a very simple body plan compared with the majority of the bilateria. They don't have any body cavity. They do have a gastrovascular cavity, which serves both as a digestive space. Remember, there's no mouth or anus in a gastrovascular cavity, just the gut, as well as circulatory space. Okay, now, now moving into the other Lophotrochozoans. In the phylum Annelida, we've got two major subclades, one of which is the Arantia, and the other one is the Sedentaria. The Arantia consists of the crawly marine worms, crawlies, whereas the Sedentaria consists of tube-dwelling marine worms as well as earthworms and leeches. Now, comparatively speaking, the phylum Annelida has a pretty complex body plan. They've got true body cavities and they're segmented. I think the whole theme of the annelids is that they've got rings around them because they've got segments in their body plan. Now, of the 12 different classes of the phylum Mollusca, uh, we're going to be talking about three. We're not going to detail them too much here because we talk about them at some length in the laboratory. But we've got one lineage giving rise to the bivalves, as well as another lineage that splits off into both the cephalopods as well as the gastropods. For those marine biologists out there, I apologize for probably skipping some of your favorite groups of mollusks, but that's the way it's going to go. We don't have time for everybody, right? Now, mollusks have pretty complex body plans, but the striking thing about this phylum is how highly divergent they are in terms of the way that they interact with the world. Their ecologies are incredibly divergent. Bivalves are filter feeding, things like clams. Cephalopods are predators. They actually chase down and overpower prey, whereas gastropods, they're kind of like the cows of the sea. They're the snails. They graze along and eat what they come across mostly. I mean, there are some predatory gastropods as well, but we see the different kinds of mollusks as having different kinds of ecologies. Okay. The key feature of the molluscan body plan, the thing that makes them special, is the presence of a mantle. And again, I'm not going to spend a lot of time or space on that here because it's one of the things we talk about at some length in the laboratory. Okay, in the structure of the arthropod phylum, we've got a basal lineage, which are the chelicerates, Chelicerata. And Chelicerata include things like scorpions and spiders, as well as mites and some other things. But that's the most basal grouping. The other lineage gives rise to a basal grouping of its own, which is the myriapod lineage of the Myriapoda, and these are things like centipedes. And apart from the myriapods, we've got another couple of taxa, which are kind of familiar ones, one of which are the insects. I don't need to introduce those to you, and the crustacea. Both of these are pretty familiar, so I'm not going to kind of describe them. Now, arthropods, arthropods as a phylum are noted because of their jointed appendages. Jointed appendages, which means that they've got legs with joints. Okay, and insects and crustacea, myriapods, Chelicerata all have those characteristics. Uh, Phylum Nematoda. Now this is kind of an interesting group because on one level they are ubiquitous. They're pretty much everywhere you look. On the other hand, they're not very noticeable. There are a lot of microscopic nematodes. Some of the bigger ones are parasitic. One particular animal that you should be familiar with here, I'll just give this worm a name. This is Caenorhabditis elegans, which is the study organism of choice for many, many biologists. Okay, nematodes and arthropods are probably the most two successful animal phyla there are. So overall, I would say the Ecdysozoa really wins out when it comes to who the most successful animals are. This includes all the deuterostomes as well. Chordates, we think of ourselves as being pretty successful, and we are in a lot of ways. But as far as overall species diversity goes, as far as overall biomass goes, we are far outdone by the arthropods and the nematodes. Okay. In order to finish out the survey of non-chordate animal phyla of interest for this particular Bio202 class, we'll be detailing 5 groups within the Echinodermata. Okay, I'm not going to put them in any particular order. Actually, they are in a kind of an order. I'm starting at the top with the Crinoidea. Crinoids are the sea lilies. They're sessile organisms that live on the bottom of the sea. Ophiuroids are actually more familiar to us. These are the familiar brittle stars. They've got kind of a central disc, and they've got wavy arms that extend away from the central disc. They can move around, but they tend to just sit on the bottom and filter feed. And then you've got the asteroids, which are things like Patrick the sea star, and then the echinoids are the sea urchins, and the holothroids are the sea cucumbers. Now, if we look at these five classes within the echinodermata, there's kind of a range. And I guess one of the themes of the echinoderms is that they have secondary radial symmetry because they are, after all, bilaterians. They descend from the same bilaterian ancestor as the rest of all these other animals that are clearly bilaterally symmetrical. So if they have radial symmetry today, it's because they had bilateral symmetry and then they lost the bilateral symmetry. They reverted back to presumably the ancestral state of having radial symmetry. And you could tell this not only from their history and their placement within the bilateria, but also when you look at their larvae as they develop after gastrulation, they become bilateral as larvae before they settle out and metamorphose into sometimes pretty radially symmetrical adults. If you look at the five classes of the echinoderms, they range from being totally radially symmetrical. Crinoids and ophiuroids are completely radially symmetrical. Asteroids are also radially symmetrical. These are the ones that are moving around, that can pretty much move around in any one of the directions. If you look at the echinoids, which are the sea urchins, some are pretty radially symmetrical and some are actually kind of bilateral. A heart urchin, for example, has got a front and a back. And then within the sea cucumbers, you've got some pretty strikingly bilateral groups of sea cucumbers. Yeah, they're things that are like the sea apples are pretty much holothroids that are completely radially symmetrical. But a good number of sea cucumbers you would look at and say, hey, that's a bilaterally symmetrical animal. Okay. Now, one of the neat things about this group is that of all the bilaterians, this is a group that seems to have the weakest of all in terms of the collinearity of their Hox clusters. They don't seem to have their Hox genes occurring in the same normal sequence of anterior to posterior that you see in the rest of the bilateria. This is almost certainly related to the fact that these guys have weak bilateral symmetry at best, only larval, or maybe even for sea cucumbers. You look at a sea cucumber and say, yeah, it's bilateral, but it's not nearly as bilateral as, say, a fish. You don't have that really strong directive dorsal ventral axis. All right.